Okay, now let's have some science. So let's introduce our science next. So please, if Kate Delfosse will come from Deep Genomics and introduce our first speaker.
Here we go. Hi, everyone. Thanks so much. I have the pleasure today of introducing our keynote speaker of the day. Dr. Clare Turnbull is a professor of translational genetics in the Division of Cancer and Epidemiology at the Institute of Cancer Research. Her research spans statistical, population, and public health-related analysis of cancer epidemiology, genetic cancer susceptibility, and implementation of expanded genomic testing. Claire has a particular interest in testicular and breast cancer. She is currently rolling out a large CRUK-funded Catalyst Award called CanGene CanVar, data resources, clinical and educational tools to leverage cancer susceptibility genetics for early detection and prevention of cancer. As part of this, she leads CanVig UK, a national and multidisciplinary network of more than 300 NHS diagnostic laboratory scientists, clinical geneticists, and genetic counselors, fully representing the 24 NHS genetic centres of UK and ROI. CanVig UK has developed CanVar UK, a digital platform for national multidisciplinary management of resources and classification for more than 2 million variants in 110 cancer susceptibility genes. Having trained as a clinical geneticist, her work at the Royal Marsden NHS Foundation Trust focuses on management of patients and families with genetic susceptibility to cancer. With that, I'd like to thank Claire for coming today and welcome her to take it away.
Thank you very much for that introduction, and I slightly wince. I think I obviously should have made it more succinct what I sent you. So anyway, that was a little bit more detail about me than you wanted. But anyway, thank you very much. So I'm going to talk today about conundrums and challenges of clinical variant interpretation, and I hope that what I say in this privileged first slot will give you a nice foundation upon which all of the amazing data and assays and results that you hear about over the next 2-4 days to land. And also it's a sort of it's a shout out for how much we in the clinical world need your assays and data. So those are my disclosures, and I am going to speak about 5 areas. So I'm going to start with a little overview about what we do in clinical genetics. So clinical genetics as a specialty emerged in the 1980s, in part because we didn't yet have the internet and you needed some doctors who had the knowledge in their heads and were able to own the books with the pictures, and also that we were beginning to map a few key diseases and have emergence of some genetic technologies, in particular microsatellite markers and linkage, which would allow us to start using genetic testing in patients. So the sort of early paradigms of those early days of clinical genetics were Huntington's disease, doing predictive testing, prenatal testing for diseases with maps, such as Duchenne's muscular dystrophy, and the beginning of this discipline called dysmorphology, seeing children with intellectual disabilities, congenital abnormalities, and beginning to group and split these into syndromes empirically from looking at the kids and from looking at the patterns in the family. So beginning to figure out the inheritance. Was it dominant, recessive, X-linked? Is it a teratogen? Is it a field defect? Largely with absolutely no molecular basis at all. And indeed, for many of these conditions, we still don't have the molecular basis. In part to give information to parents, but often actually to give a recurrence risk about a future pregnancy. However, that was the 1980s. We now, due to the revolution in genomics and much more wider analysis and research, we do have the genomic basis for many of the diseases that we were historically seeing empirically. And now we can do a little bit more. We can give people a result and manage them. So in rare pediatric disease, what does that mean? Well, it means that we can end the diagnostic odyssey. We can give a bit of a prediction about the future. We can maybe know what we need to monitor for, what we need to keep an eye on the horizon. We have drugs for a few of these conditions, other drugs in trial. But again, a lot of it is around reproductive options. So pre-implantation genetic diagnosis, so that's going through IVF and embryo transfer, selecting the embryo based on the genetic mutation in the family, or prenatal testing, so that's having termination of pregnancy based on finding the specified mutation in the fetus. In cancer susceptibility, where I work, we have a broader palette of management options. If we know someone's at high risk based on their predisposition mutation, we can offer them enhanced screening, which is not economically viable to offer to everyone in the population. But for example, our BRCA carriers, we give them annual MRI from age 30 every year, whereas the rest of the population get mammograms from age 50 every 3 years. We can offer well people drugs that have side effects to mitigate the disease of which they are at high risk. So we use aspirin in colorectal cancer, in Lynch syndrome, tamoxifen, which is an estrogen blocker with a relatively unpleasant side effect profile, we use for our BRCA2 carriers. There are a few targeted drugs coming down the pipeline. There's a trial of denosumab in BRCA1 carriers, and rumblings of a PARP inhibitor trial. We can recommend behavioural interventions, which again would not fly with the general population, but if you're at very high risk, you may modify your behaviour. Classically, CDKN2A, very high risk or elevated risk of melanoma, so those guys would want to modify their behaviour. And then we can offer surgical, so preventative surgery, removing healthy organs which we understand to be at elevated risk of disease. So classically, we would offer mastectomy or oophorectomy for BRCA1, BRCA2, but we do offer preventative colectomy for our APC mutation carriers and gastrectomy for CDH1 mutation carriers. And I just emphasize that this is on the basis of our interpretation of the mutation, and those are some pretty binary, dichotomous, irreversible clinical decisions we take. So what are the challenges here of this clinical genetics paradigm? Well, these diseases we're managing are pretty rare. The effects may be a long time down the road. Therefore, the clinical management has emerged in time. It's quite heterogeneous in space. We've changed it over time, and this means that actually our evidence base is pretty limited as to being very confident that the interventions we're offering are genuinely impacting on the outcome, impacting on genuine survival and measurable outcomes, let alone the health economic basis. And almost invariably, our modelling and analyses around clinical efficacy are based on a pretty dichotomous paradigm of those with the pathogenic variant are at high risk of disease and those without it are at minimal risk. And that is the presumption upon which we base our management. So that takes me nicely through to my next area, which is phenotype and ascertainment. And I'm just going to grab my glasses. I can't quite see the screen. I'm squinting at it. Right. There we go. Right, that's better. Phenotype and ascertainment. So, what do we mean by phenotype? Well, in this context, the phenotype is the constellation of manifestations that we ascribe to that germline mutation. And as I said, typically it may be a range of elements to the phenotype. And I just want to think about two sort of paradigms, the significance of those features and how we ascertain them. So you can sort of separate the features into two groups. The ones that are important, that's why we care about making that diagnosis. That's why we're looking into it. So it's the intellectual disability, often in pediatric paradigms. And in my world, it's the invasive cancers. But there are also then other features which are fairly innocuous, don't have much impact on the patient, but they're often the sort of, we call them hard handles. They're the more specific, rare features by which we make the diagnosis. So often again, it may be dysmorphic facial features, it may be peculiar skin changes. And then thinking about the ascertainment of the features. So there are features that you would see walking down the high street. So short stature is apparent to all. Intellectual disability, to some extent, again, is manifest, the invasive cancer. A lot of the features that are part of our phenotypic description, you will only find if you go looking for them. So here are a few favourites of the clinical genetics world. We've got axillary freckling. Most patients don't know they have freckles in their axilla, and a physician is only going to find it if they have a look. Underneath that, we've got so-called ash leaf macules, tuberosclerosis. So these are hypopigmented patches. So if the patient has dark skin, you can see them reasonably well. But if they're pale, you literally have to shut that child in a cupboard with no light and then get this thing called a Woods lamp and then flash it in order to find these patches. Chirpy is, again, an innocuous change in the back of the eye, which is quite specific for FAP, for APC mutations. Again, you will only see that if A, you have an ophthalmoscope, and B, you know how to use it. And then this is a classic jaw cyst, CDC73, hyperparathyroidism, parathyroid cancer. So there are these so-called sort of pathognomonic cysts on the OPG, which literally you have to go down to radiology and beg for. That is the only way you'll get one of those films. So again, this is just not something you kind of, it's biting you on the nose in regard to phenotype. So, bearing that in mind, I'm just going to describe a couple of syndromes that we see. Rubinstein-Taybi, CREP-P mutations. So why are we interested? Because it causes reasonably severe intellectual disability. These kids are short, but how do you make that diagnosis in clinic? The so-called distinctive facial features. So they have a pinched nose, downslanting palpebral fissures, and these distinctive broad thumbs and toes. So that's a classic sort of dysmorphology syndrome. In the world of cancer susceptibility, we love looking for Cowden syndrome caused by P10 mutations. Why do we look for this? It's associated with elevated risk of thyroid, breast, endometrial, and colorectal cancer. All of those are pretty common. So what are our hard handles? They are these skin changes. So these tricholilomas, which are essentially, as far as I can, sort of like little bumps that you get on your face and your neck. There's also some slightly different bumps called papillomatous papules that you get on your hands and your feet. Something called a cobblestone tongue. And what's slightly more objective is macrocephaly, but you have to measure the head properly. And then there are different charts and scales which varies by age, obviously, but different charts for different sexes, different charts for different ethnicity groups. And actually, there's quite broad familial variation, which is autosomal dominant. So those are a couple of sort of classic syndromes we would describe in clinical genetics. So what are the sort of complexities here? Well, these are the features which we use to weigh up how we interpret the likelihood of a vast and uncertain variant being pathogenic. So in your world, this is what we're using to weight our certainty of pathogenicity from the clinical perspective. And as you can see, these features are all very subjective in how you describe them. There's an absence of systems for really quantifying, a pinchy nose scale. These things don't exist. We're very, very poor at documenting them. HPO is a sort of ontology, but it contains very little subtlety to it. Many of these features only come on later in life, so those skin changes of Cowden's will only be sort of manifest around about age 30. And we have zero good evidence of the frequency of these features in the general population. We have not done an auxiliary survey of the population for freckling, so we don't really know the level of specificity. And this really has been a significant problem up until maybe 2, 3, 4 years ago, because we did single gene testing, and you could only get that single gene test if you justified that the patient had the thing that you were looking for. And therefore, you had an enormous bias towards conformational reporting bias, that once I thought that their face looked a bit like Rubinstein-Taybi, I'd be going, look at those broad thumbs, guys. Those thumbs are broad, aren't they? Or I'd go off doing my X-ray or my biochemical test because I'm chasing confirmation of my starting presumption. You also have ascertainment bias. I can only do that test in people who have the tick box of things that we think go with that syndrome already, and ergo, well, yes, we then analyse that data and find that these features are strongly associated with the gene. Over the last 5 or so years, we've done much wider agnostic testing clinically. So rather than doing single gene tests for our kids with intellectual disability, more typically doing a broader test, and often a whole genome or a whole exome. And actually what that means is that you test all the genes agnostic to phenotype, and you start finding your CREB-P mutations going, those thumbs look completely normal, as does the facial morphology and so forth. So that's very much sort of unpicking a lot of the associations and also doing a lot of whole genome, whole exome sequencing in the general population. If you can then link that back to phenotype, so for Biobank, they can link it to the cancer registry. And there have also been various studies of secondary findings, so actually going back to the patient and getting a personal and family history. And again, a lot of our cancer associations, which we have ascribed to these genes, are less strong than we originally presupposed based on our ascertainment bias. So my third theme is variant-specific risk, the entity of which we dare not speak. And I will start with protein truncation variants. We thought we were safe with these. I mean, they cause nonsense-mediated decay, so surely they must all have the same risk. And there's actually BRCA1 and BRCA2. This entity was described first in 2001, that actually truncating variants in the center of each gene have a disproportionately high risk of ovarian cancer and low risk of breast cancer, and truncating mutations at the peripheries of the gene have an elevated risk of breast cancer and a disproportionately lower risk of ovarian cancer. And this was first described in 2001 by Doug Easton in the Breast Cancer Linkage Consortium, and then replicated in a much larger data set in 2015. Clearly, BRCA1 and BRCA2, we've got large numbers of ascertained probands and families and good epi data, but it's quite plausible that this phenomenon also is true for many other genes. And it may be that this is due to leaky splice sites and isoforms around sort of consensus splice, canonical splice residues, or it may be true actually for all nonsense mutations. So I think that is food for thought. Empirically, missense mutations we would think would have different risk based on their location in the gene and also the nature of the substituted amino acid. There are a handful of sort of well-documented, proven instances of this based on clinical data. So R1699Q in BRCA1 is a relatively recurrent rare BRCA1 variant. So Mandy Spurdahl collected together enough families that they could do segregation analysis, which would then demonstrate that this variant does confer risk of breast or ovarian cancer, but it's statistically significantly lower than the risk of a standard BRCA1 mutation. There's an ATM mutation, V2424G, which again it's rare, but it constitutes about a third of pathogenic mutations in ATM in Western Europeans. So they could collect enough families, again, to do segregation analysis and actually subsequent case control analysis. So this variant has an odds ratio for breast cancer of about 8 compared to an odds ratio of 2 for classical ATM mutations. And then this RET mutation, V804N, we tripped over this actually looking at a clinical case a few years ago and noticed they'd just re-released EXACT without the TCGA element. And there were 11 instances of it, which just seemed too many. So using the MTAF principle, the maximum tolerated allele frequency, which is basically a sort of fundamental inherent relationship between the frequency of your phenotype in the general population, the frequency of your variant in the general population, the attribution of that phenotype to your variant, and the penetrance. Those are a sort of four-way ipso facto. We calculated this variant was likely to have a penetrance for medullary thyroid cancer of estimated 4% with a confidence interval of 1 to 8%. And actually, we manage inherited RET mutations by a preventative thyroidectomy in childhood. So this is very important. It's based on a presumption of very high penetrance, and clearly this mutation, which is a long-standing documented one, is much lower. So clearly, variants do have different risks, but they will have individually different risks for each element, each subphenotypic element, which is a level of complexity you don't really want to think about. It is a problem to quantify these risks clinically because the variants are individually very rare. It's just very difficult to collect together enough probands and families. A lot of the phenotypes we deal with, they're not quantitative, like, I don't know, height or enzyme level. They are binary. So the data collection and analysis just requires that much more power. But of course, going back to my first theme, it's really important because our clinical actions are based on a presumption of sufficiently high risk of the variant that your benefits from the clinical actions outweigh your harms. And I think this is an area where potentially MAVE data can really address the issue. Of course, the problem is you need enough clinical true positives to calibrate your intermediate effects on your MAVES assays as being accurate. And also then you will need to do enough replicates of your MAVE assays that you can be confident that those intermediate values are genuinely reflecting intermediate clinical risk and are not just a sort of a sampling variation from your other results. So my fourth theme I want to speak about is variant interpretation frameworks. So how do we come up with these magical decisions that a variant is pathogenic? Well, in the good old days, we say, I've seen that variant in a clinical case that has the right phenotype. So therefore, that variant must be pathogenic. And that was really roughly, you might, for belt and braces, check if it's deleterious on SIFT. But ultimately, that was roughly how it was done in the early days. And I was pleased to actually unearth this slide, which I must have made in 2011. So we dumped out HGMD and got all the variants in HGMD for BRCA1 and BRCA2 in November 2011, and there were 936, of which roughly 3/4 were DM and 1/4 were query DM, and DM means disease-causing mutation. And cross-validating at the time, various of them were sufficiently common that you knew that they were benign. But HGMD was a hand-curated database of clinically important variants reported in the medical literature, but their modus operandi at that time was any rare variant reported in the case of the appropriate disease was ascribed as DM. And obviously, was overall a flawed strategy, this is particularly flawed for any phenotypes that are pretty common, like breast cancer. And this is a particular problem because actually in that era, the computational biologists thought that HGMD was a sort of, a bona fide clinical truth set. So we're using HGMD to then train various in silico tools and so forth. About the same time, at the other end of the spectrum, there was an emergence of multifactorial tools. So this was a real landmark paper in 2007. This is, again, Doug Easton working with David Goldgar. And this was in the good old days when Myriad shared their data. So Myriad, when they did the BRCA1/2 test, also if they found a rare variant, free of charge, cascaded out to the families. They had a fantastic data set, also good clinical data with that. So Doug and team took data on several 100,000 BRCA1/2 tests, and they did a kind of logistic regression that weighted on phenotypes. That's your first column, the family history log likelihood ratio. They then looked for co-occurrence in trans with an established pathogenic variant, which would be evidence against pathogenicity, and then got another log likelihood ratio for co-segregation. Add these up, so you get a summed log likelihood ratio, and they could then flip this into an odds of causality or odds of neutrality. And then this was a sort of starting basis for these types of multifactorial analysis, upon which they then added in a prior based on conservation and wove in some sort of tumour data for another phenotype, and then started bringing in functional data as well. These, very, very good variant classification, but only undertaken, to my knowledge, for BRCA1, BRCA2 and Lynch, and even only for a relatively modest number of variants therein. So the American College of Medical Genetics, recognizing essentially that variant classification was somewhat of a wild west with occasional exceptions, in 2012 convened a group to develop a framework. And I'm sure everyone in the room is familiar with this sort of now iconic rainbow grid. But this contains 27 evidence items, which are given a score of either supporting, moderate, strong, or very strong towards pathogenicity, or supporting or strong towards benignity. And they're grouped into different themes. And then there's a sort of scoring system where if you have a grapefruit and a cherry, you can get pathogenic. If you've got 2 kiwis, you get likely pathogenic, and so forth. And actually, this has been essentially universally accepted, which is fantastic. We are all now using the same variant interpretation framework. And the authors themselves at the time said this is a starting point upon which you as a community need to build. And one issue with it is that the terminology is deliberately quite loose and descriptive. So it's a well-established functional assay. Evidence of statistically higher number of variant in cases than controls, and so forth. So the ClinVar SVI, Sequence Variant Interpretation Group, sort of oversee this specification. And then there are VSIPs, which are Variant Curation Expert Panels, so essentially per gene experts go away and sort of take away the ACMG and do it for their gene. We have a group in the UK, CanVig UK, so in 2016 it was agreed by the UK Association of Clinical Genomic Scientists that we would adopt the ACMG framework nationally. So we set up 2 groups to sort of educate and specify how we would use the ACMG framework across the UK. So I lead CanVig UK. We've been going since 2017, so we have each of the labs represented, and we have at least 1 clinical scientist and a clinical geneticist from each lab. We meet monthly. We review emerging guidance from the different VSIPs or the SVI, and we actually, because there's still a lot of gaps, we pulled together our own guidance to make us consistent nationally, and we keep that updated on our website and keep adding to it and amending it. We have educational presentations. So a lot of it is quite tricky and needs unpicking. And we also, each month ahead of the meeting, send out variant of the month for the lab scientists to classify, and then we review it in the meeting. And it's actually very helpful because we pull together not just the final score, but how they got there, and use that to unpick variation in practice and understanding of the ACMG. We also have CanVar UK, which is a platform by which we can share our, you can send out to the whole network. And we are actually now going to open this up shortly outside of the NHS. But so within CanVar UK, it contains every possible variant in these genes. So you can send out an email that then has the variant as the header, with your query, and then anyone can just push that button and log in, and then it means we kind of document these conversations, which until we got this system essentially were just stored in a bunch of emails. So we do now have a single place where we keep all of our discussions, and then if, as and when we as a group make a consensus classification, we then submit it to ClinVar. Second issue, the ACMG framework, the evidence in there is these evidence items are not independent in some cases. So again, we, within our CanVig, we keep a kind of fairly didactic sort of rules about what you can and can't combine and when you downgrade, which is agreed. Some of it is somewhat arbitrary, but at least we are trying to be consistent. And I know that that's a big area the ACMG update is looking at. A third issue was that the ACMG is sort of categorical, and that kind of 2 kiwis or a grapefruit and a cherry it didn't cover every possible combination, and there was a sort of level of frustration. And also at the higher end, it was fairly inflexible about being able to weight the evidence. So Sean Tavtigian in 2018 sat down and essentially sort of figured out, and it's quite straightforward, that you could convert this with relatively perfectly, there are a few little outliers, into a Bayesian framework. So you presume a starting prior of pathogenicity of 10%, and then from that you can make your points into likelihood ratios, which are related to each other multiplicatively at log to the base 2.08. We in CanVig liked this, so made it into a scoring system, And the lab scientists prefer this because it's a lot more simple and objective how you combine the evidence items. And likewise, Sean also sort of published on converting this into a simple scoring system. And I understand there will be an updated framework from the ACMG, which is likely to be much more points-based and allow you a bit more flexibility around getting allocating points between that kind of 4 and 8, which was pretty inflexible. And then finally, again, something which I think is a sort of iterative approach, that you had your VSIPs, your gene-specific groups, were defining some sort of universal evidence items, I don't know, about truncating variants or in silico tools. They were kind of all defining them quite differently. And they were also using quite different weightings for their phenotypic evidences. Some of them are quite punitive and some of them are quite generous. So again, I think that will be requiring sort of evolution. But essentially, the ACMG framework, big step forward. It does need to continue work in those areas that I've discussed around the sort of non-independent items, how you sort out gene-specific versus universal. I think there is still a tension in certain groups. Don't like this is a sort of devolved framework, and the anticipation is that the lab scientists out there do it themselves. There are some groups who would prefer to own the variant classifications and release them, and that they should be sort of taken as a sort of predefined classification. I think that tension is not solved. Ultimately, it's actually still quite hard work. There are 27 evidence items. A lot of them are, with the best will in the world, manual. You have to go and look them up in different places. You have to decide how you weight them. You have to go to all your different evidence sources, go to the supplementary materials, download the PDF. This is really time-consuming. The lab scientists spend a lot of time doing this, and ultimately, it's still quite often inconsistent, as I showed in our CAMVIG survey each month, and also if you look on ClinVar. So I'm now just going to speak finally about a sort of bringing things together, a few thoughts about clinical interpretation and MAVES and what we need to do. So there are all our sort of nebulous variant classifications. There is Mother Nature's truth, which she hasn't told us, but that is what we are trying to hone in on. And there's evidence I think it does sort of map to 3 quite clear areas. There are observations around that variant in individuals with and without disease, so various types of evidence relate to that. There are functional ex vivo variant-specific assays, which is what you guys are doing. And then there is data from the inherent genomic context or computational prediction. So just talking a little bit more around what the clinical community need to do. So as I said, in the bad old days, we overscored phenotypic specificity. We thought that was enough to ascribe pathogenicity. You're over the line. So the ACMG came out and said, right, slap on the wrist. You can only have supporting for phenotypic specificity. PP4, you just get 1 point. And the truth is somewhere in between, but it is quite specific to the context. It's specific to how specific that phenotype is, how predictive it is for that gene, and how constrained the gene is. And we tried to come up with a way of quantifying this. So this is Alice, who works with me. So we called it the very rare missense variant likelihood ratio, which is quite a catchy name. But essentially, Alice, we used pheochromocytoma and paraganglioma, which is a rare type of endocrine tumor, as our exemplar. Alice compiled over 6,000 cases who had had testing for SDHB or SDHD, which are the key genes, which involved getting data from the 2 big labs who test those genes in the UK, all of the French data, all of the Ambry data, thank you Ambry, and TCGA, and then could quantify a likelihood ratio for the likelihood of seeing a very rare missense variant and it being a case versus a non-case. And we used a likelihood ratio because that then slots into the ACMG. So that likelihood ratio is about 75 for all rare missense. And even if you get rid of the ones that we have already classified as pathogenic, so these are now sort of unknown missense, you're still at about 35. So that's quite helpful. You can then advance that by looking in a domain-specific way. So again, certain domains are even more predictive of pathogenicity. And then we also looked at various subphenotypes, which again can boost your likelihood ratio. So if it's head and neck, if it's invasive, if it's multiple tumours, if it's familial, and so forth. And this is something, in principle, one could extend to other phenotypic paradigms. So to that end, over the last 5 years, I've been working quite a lot with a big project funded by Cancer Research UK, working with Public Health England, which has now become NHS Digital. So we now have all 18 English labs submitting individual level data on testing of cancer susceptibility genes into a secure data environment within NHS Digital. So we're doing pretty well. The data's been coming in since 2018. These are people who have had a diagnostic proband test by NHS eligibility. So they have a good going phenotype to earn their test. We get submission of all testing, so we get the normal and the abnormal, which gives us a denominator. The labs were given a common data model against which to submit. And the labs can variously go back, but some of the labs have been able to go back to to give us their full historical testing back to 1999. So this is a great data set. They have an NHS number. We can actually link these individual level data to a ton of other things and find out about their chemotherapy, their survival. So there's lots of other use cases. But for the purpose of variant interpretation, we want to collapse down the data and get a frequency with a denominator, which is great. So we can provide these frequencies back to the clinical community in our Canvar platform. So I can't see that, but essentially you get a numerator and a denominator. And then we can release the self-reported white ethnicities separately. So you get a numerator and a denominator for that, which then allows you comparison to the non-Finnish Europeans in NOMAD. So you can get a p-value, a case control p-value. So that looks great. This has been an enormous and painful undertaking because what it has revealed is that all the labs use completely different LIMS systems, laboratory information management systems. Although we gave them a common data model, they all gave back their data in completely different formats, completely different structures, different contents in different fields. So we then, Francesco needed to write these individualized scripts, algorithms to remap the data, extract bits out, put those together. Most of the actual variant data was submitted within a word salad of free text. The phenotype data were very poor. We only knew what the phenotype was on the basis of the genes that had been tested and so forth. So collecting phenotype data in cases is pretty hard work. Another area which we've been working on is trying to think about how we use data from one area to quantify the predictiveness of a tool or a test or a resource in another area. So Jean-Kut in my team was sort of experimenting with this, looking at using some assay data. So we took 5 big assays to quantify the predictiveness of in silico tools. So again, there's lots in here. And essentially you can see by that big red bar, that's all the true positives. These tools are all pretty good at calling deleterious variants. But to the right of the big red bar, they also like to overcall benign variants as well. So we looked at various metrics. We looked at balance accuracy against the 5 assays, individually and in combination, also against ClinVar, but essentially coming out for balance accuracy was Revel and Metasnip. So we then again looked at these at different scoring thresholds to look at likelihood ratios. So again, try and quantify how predictive these tools are, and particularly at different scorings, either high or low, for these different in silico tools. So there's lots more in that paper, but again, that allowed us to quantify what these tools are telling us using the MAVE data as a truth set. Likewise, PM5 is an evidence item that you can get moderate evidence if your variant under examination is in the same codon as a reported pathogenic variant. So we wanted to really quantify, was that about the right level of evidence? So we used the Sterita Finlay BRCA1 data and the Kitzman MSH2 data as our truth set. We also use ClinVar, but you can imagine that's actually a pretty bald, sparse truth set for this type of analysis, and again could quantify in likelihood ratios how much that can within codon concordance was telling you, particularly if you set rules like your variant under examination has to be more deleterious with this tool, or what happens if there are 2 pathogenic variants also at the codon, and so forth. So again, using a data source as a truth to quantify the predictiveness of another tool or data item. So I'm going to pull together now and finish off. So, phenotypic evidence, my guys, the clinical people, we need to do a lot more, because we need to improve our classifications, but we also need to provide good truth sets for you guys and other people who want to then cross-validate their assays or tools. So we need these large individual level data sets. We need them complete so that we have a denominator. We also need good phenotype data, and that is what is so hard for all of the many reasons I've alluded to over the last 40 minutes. I think we need to be able to amalgamate these data sets across different suppliers. So again, shout out to Ambry, who are very good at sharing data, but ideally it would be great to combine our NHS data set with multiple different clinical data sets, but it's that individual level and phenotype data that you need to be able to combine. You guys, functional assays, until very recently, it was pretty infrequent that one could use those, one would have recourse to assay data for a classification, and often you didn't really know how to use it. Often the assay may not correlate with clinical pathogenicity, they were often non-reproducible, or only assayed, a handful of variants, so it was unlikely your variant you're interested in would be there. But actually now, as you are generating large, comprehensive assay data, I think we clinical guys are hoping that this is really going to sort of clear up the field and help us move forward. And then again, as a broad community, we need to be intelligent and canny about how we iterate off each other's data to make truth sets so that we can hone in on Mother Nature's truth. And I think we need a methodology to how we do that, because as part of that, we will evolve a sort of ever-improving truth set where we can kind of pick out the miscreants and keep improving. And I think that the ACMG framework is a very good universal structure which helps us be consistent along with these other structures from ClinGen and ClinVar. So I shall finish there. Thank you very much for listening. I've spoken about clinical genetics, how we manage patients. I've spoken about phenotypes and ascertainment and all of that bias, variant-specific risks, which we don't want to think about, but we need to, variant interpretation frameworks and the sort of historic overclassification, and about how the MAVE world and the clinical world, we need to have good ways of bouncing off each other and working together to improve our classifications. So just a thank you to all of my team and people who contributed.
Yeah, so I realize that BRCA work on sort of where the mutations are leading to different outcomes is not yours, but it's so interesting, this sort of divergent mechanism. I was wondering, what is known about what follows that and how that plays out in different types of assays?
I have no idea. Yeah. And to my knowledge, there isn't an explanation, and that's still, although I kind of included it under variant-specific effects, that's still pooled effects. And it was a statistical model of sort of trying to best model where those risks, where the tipping points overlapped. So I don't think anyone has a specific hypothesis, but it has been reproduced and it's reasonable numbers. But you're not only collapsing the variants by region effectively, they're all collapsed by mechanism as well. And I guess looking back from 2022 on those data, saying actually I'm not sure if you've included some very leaky splice variants, and that might be in some way influencing it.
Right, that like some is an RNA mechanism and some is a protein degradation mechanism. And,
But they they are showing different risk between two different phenotypes. So there is some phenomenon there, whether you can sort of question what's driving it. I don't have any great explanations.
Very cool. Thanks.
Actually, there aren't any online questions yet, but I will take that opportunity to ask my own. So when we're designing our studies, how can we best design them such that we can convince clinicians that our data sets are of high quality? So other than just comparing them to gold standard sets, once we're finished, are there things that you look for systematically when you're looking at our data sets?
I mean, I think that's almost like a sort of like you're a plant for why the CVI group is useful, because I think if you can have sort of agreed ways of annotating the variants and then converting annotating the variants, sort of signing off the QC metrics and then converting them into meaningful scores, I think the clinicians, and it's really the clinical diagnostic scientists, are very, very happy to use your data. They want to use your data, and they just need some recognized sort of system of validation and approval to give them the go-ahead.
Thanks.
Hi. So we're going to hear from Debbie Marks later today, right? And it seems like there's a few papers that have come out suggesting that computational variant effect predictors may be able to provide somewhat stronger evidence than supporting evidence. And so I'm wondering, we've been thinking a lot in our group about how that's going to play with functional data, and whether we can convince ourselves that what types of predictors are really actually independent of what types of functional assays, and how they could be combined. Is that something you all are thinking about also, or is it a naive question?
I think it is that sort of iterating in on the truth, but recognizing the potential collinearity. And again, I think I can articulate the problem, but I don't have a specific solution. But I think it is sort of constantly reintegrating, reanalyzing, and looking for the outlier. But it almost feels like there is a methodology there. And if we could all feed into that as a longitudinal process. Yeah, but more specific than that, I cannot offer.
Okay, thanks.
Hello, thank you for your talk. We are seeing more patients nowadays having whole genome or whole exome sequencing. I'm wondering, based on your knowledge, what do you think is lacking now to have this shift from single gene testing to large-scale panel testing? Is maybe we are just waiting for functional data, or maybe has something related to eligibility for insurance to get tested or something like that?
I mean, I think there are lots of questions and themes within that. I guess for research, it's useful to help us better understand these genes, to collect good phenotype data on the patients, and that's often lacking, particularly when the sequencing is provided by a third-party commercial provider, because there's often a disconnect, and it's as much often the clinician's fault for not supplying the data. To do the whole exome in the genome, and if we have those data, we can then do better analyses to understand penetrance and modifiers and so forth. From a clinical perspective, it's often useful to actually ask a specific question, because a whole genome and a whole exome, it's asking 20,000 questions, and our ability to understand those answers is quite limited. And actually there's the syndrome without a name, the SWAN. I think, in a sense, the new era has slightly set us back, because we used to have quite tight understanding of what happened to our patients. It might have been wrong, but it was tight, because we tested patients who looked like they had the thing we were looking for, and we had a bit more understanding of what happened to those patients. Whereas if you test without any starting presumption of phenotype, you're getting the patients who are a little bit further along the sort of distribution of modifiers, and they don't behave quite like the patients that we've previously studied. So I think, yeah, we're now in a sort of era of having less secure understanding in terms of prognosis and outcome and penetrance. And in particular, you know, when that comes to predicting risk in future pregnancies and so forth, I think that it's becoming increasingly challenging.
Yeah, I know that Canada offers single gene testing for free for people that are in high risk, but you also get an offer, like a panel of genes from a private company, everything, just the question of what would be actionable.
Yeah.
Thank you.
I mean, there's certainly been a commercial drive to testing larger panels of genes or doing larger tests, and that looks like you're offering more, but it does sometimes mean that the results we find, we're on less stable ground to offer interpretation and prediction of them.
Thank you.
Thank you very much.